Good morning children, I am Segregation. Now I am going to teach you the principles of inheritance. Today we can see monohybrid and dihybrid cross conducted by Gregor Johann Mendel on Pisum sativa. Monohybrid cross I taught you, that is the plant breeding which is dealing with only one character at a time. In his experiment, Mendel was selected one tall plant and a short plant. That means height he was considered as the single character. So he was selected one tall plant and a short plant. One male and other female. And in the first generation, so this is a parental character. So this is, we can call it as phenotype. So tall and short are the phenotypic characters. Phenotypic characters of the P generation. What is P generation? Parental generation. So this is the phenotypic characters of the P generation. And in his first generation, after hybridization, he got all tall plants. Okay, in the first generation. So this is called the first filial generation. You already learned in 10th standard. So first generation is known as filial. The generations are known as filial. So F1 means first filial generation. Here, all the first generation were tall characters or tall plants. And he conducted self-pollination of the same generation. So, male plant is also tall and the female plant is tall. Then in the second generation, he got tall plant and short plant. But the ratio is they show variation, that is only 75% tall and 25% short. So, not all the plants were tall. 75% of the plants were tall and 25% short plants. And from this experiment, he concluded that in the first generation, the offspring show one of the parental characters. Which was the parental character? Tall character. So the first generation offspring were showing one of the parental characters, and that was tall character. But in the second generation, the offspring showed both the parental characters, right? That means the character which were hiding in the first generation expressed in the second generation. Here, the shortness was hiding in the first generation, and that will be expressed in the second generation, and that is the short character. These are monohybrid crops you already learned in 10th standard. Anyway, we can see once more. Is it clear? So, the tall character and short character were hybridized together. In the first generation, he got all the tall plants. That means the offspring show one of the parental characters, and the next character was hiding here. And in the second generation, the offspring show both the parental characters. The character which were hiding in the first generation is expressing here in the second generation. That is a short character. But only less number of short plants he got in the second generation. And from this experiment, he concluded that something is transmitting from parents to the progeny. And he gave the term for that unit of inheritance as factors. And later it was given the term genes by Johann. Okay. Now we can see how he had given letters to this characters. See, as I told you, the tall and short are the phenotypic characters. Am I right? The phenotypes. So here, in the first generation, all plants were tall. That means which is the dominant character? Tall is the dominant character, and the recessive character is short. So he was given capital letters for the dominant character. Okay, so capital letters T capital T, as the genotype of the tall character. So this is the phenotype of the parents, and here genotype of tall is capital T, and he was given small letter, the same letter he was given, not S letter, the same letter, small letter he was given for short character. Okay, so to avoid confusion, he was given the same alternative letter or small letter to the short character, that is small t, that is the genotype. Why he was given capital T to the tall character? Because tall character is the dominant character in the first generation. You must notice that the tall character is the dominant character in the first generation. That's why he was given capital letters to the dominant tall character. And for the recessive character, he was given small letters. Okay. And now we can see, so these are homozygous alleles. Here also homozygous alleles. So the homozygous alleles always produce a single type of gamete. For example, I will give you another example. You know the chromosomal constitution of female and male. So this is an example, as an example, and this is the chromosomal constitution of male. So here, due to this homozygosity or similar alleles, they produce only one type of gamete, that is X. You already learned. And this is heterozygous alleles or dissimilar alleles. So because of that heterozygosity, that produce two types of gametes, which are the X and Y. When this X fused with the female ovum, that will be a girl child. When the Y fused with the ovum, that will be a boy child. You already learned. So that means the homozygous alleles always produce a single gamete, single type of gamete or only one type of gamete, but heterozygous alleles produce two types of gametes. Is it clear? Now we can see here, this is a homozygous allele. So only one type of gamete produce. Gamete we can encircle. Here also, due to homozygosity, they produce only one type of gamete. So this is the gamete. Is it clear? So the gamete fuse together during fertilization and then form. So he noticed that the first generation was tall, all tall plants. Is it clear? So this is the first generation, F1 generation. F1 generation were all tall plants, but the problem is here the genotype is entirely different from the parents. Actually, the F1 generation show the parental character such as tall character, but genotypically which is showing variation. Is it clear? Phenotypically it resembles the parent, but genotypically it shows variation. Is it clear? So now he did self-pollination when F1 cell, that means take the same male and same female. That means tall plants, one tall plant with the genotype capital T, small t, and one female plant, female tall plant with the genotype capital T, small t. Okay, so tell me, this is the parent or P generation. Tell me the gametes, capital T, capital T, small t. That means due to heterozygosity, they produce two types of gametes. Is it clear? Here, due to homozygosity, they produce only one type of gamete. Here also, two type of gamete. These are the gametes. Okay. Now we can see. Okay. Here, this capital T fused with the capital T to form capital T, capital T. Here also fused with the small t, capital T, small t. Then here also, so we should write the dominant character first. So capital T, small t, then small t, small t. So all these are genotype. So we got the genotype only. So we have to find out the phenotype. Okay, tell me the phenotype. So this is tall, am I right? And this is tall, but the problem is this is a heterozygous tall. This is homozygous tall, this is heterozygous tall, and this also heterozygous tall, and this is short. So this is the phenotype. Is it clear? So here we got one homozygous tall, two heterozygous tall, and one homozygous short. So can we find out the phenotypic ratio? Phenotypic ratio. How we can find out the phenotypic ratio? Okay, let's see. Now we can see the phenotypic ratio. Phenotypic ratio means, so here which are the phenotypes? Tall and short. How many tall? 3 tall and 1 short. So we can write 3 tall and 1 short. So phenotypic ratio is 3:1. Okay, let's see the genotypic ratio. So for genotypic ratio, we have to find the genetic makeup. That means 3 tall means here genetically some differences are there. That means here this is a homozygous tall and these two are heterozygous tall and these short gland is homozygous. So we can say one homozygous tall is to 2 heterozygous tall is to 1 homozygous short. So this is the genotypic ratio. So genotypic ratio is equal to 1:2:1. So is it clear about the phenotypic ratio and genotypic ratio? So what is the phenotypic ratio of a monohybrid cross? The phenotypic ratio of monohybrid cross is 3:1 and the genotypic ratio of monohybrid cross is 1:2:1. Is it clear? So in this experiment, Mendel was selected or considering only one character at a time, and that was height of the gland. But in dihybrid cross, he was considering 2 characters at a time, and that was color and shape of the seed. You already learned round green and wrinkled yellow. So that we can see now. So he was taken round green and wrinkled yellow. You already learned in 10th standard. Anyway, some findings only I will discuss to you. So in the first generation he got all round yellow offspring. That means the parents show variation from the parental character. Am I right? So the parental characters were round green, wrinkled yellow, but the offspring show entirely different character from the parental characters, and that was round yellow. And in the second generation he got all the parental characters such as round green, wrinkled yellow, then round yellow, along with he got wrinkled green. So wrinkled green. So one character other than the parental character, entirely different parental characters he got in the dihybrid cross. That you already learned in 10th standard. Anyway, we can see the phenotypic and genotypic ratio of dihybrid cross. Let's see the phenotypic ratio. So phenotypic ratio of dihybrid cross is 9:3:3:1. Okay, and now we can see the genotypic ratio of dihybrid cross, that is 1:2:2:2:1:2:1:2:1. So this is the genotypic dihybrid ratio. So you keep it in your mind, the phenotypic monohybrid ratio, genotypic monohybrid ratio, then phenotypic dihybrid ratio and genotypic dihybrid ratio. Is it clear? And now we can see, based on this experiment, Gregor Johann Mendel developed laws of inheritance, and that laws of inheritance was strictly based on this monohybrid and dihybrid cross. So let's see, it's very important area of this chapter, laws of inheritance. Laws of inheritance was developed by Gregor Johann Mendel based on his experiment or hybridization experiment on Pfizer sativa. And these laws of laws of inheritance consist of three laws. First one is law of dominance. Law of dominance. Second law is law of segregation. And this law is also known as law of purity of gametes. Law of purity of gametes. And the third law is law of independent assortment. These are the three laws under laws of inheritance by Gregor Johann Mendel. And out of these three laws, these two, the first and second law was strictly based on monohybrid cross. These were based on monohybrid cross, and the third law was based on dihybrid cross. Is it clear? So, the three laws under laws of inheritance are law of dominance, law of segregation, law of independent assortment, and the first two laws were based on monohybrid cross, and the third law of independent assortment is based on dihybrid cross. Let's see what is laws of inheritance in detail. First we can see law of dominance. So, law of dominance. So this is the first law of laws of inheritance. According to law of dominance, when a pair of contrasting factors of a character are brought together, one character or one allele is dominate over the other and is expressed in the first filial generation. That is law of dominance. For example, when a pair of alleles are found to or are brought together, here one character is or one allele is dominate over the other, and that determines the character. So this is the law of dominance. That means when a pair of contrasting factors of a character are found together, one allele or one gene is dominating over the other and is expressed in the first generation. That is called a law of dominance. And next we can see the next law, law of segregation or law of purity of gamete, law of segregation. In law of segregation, when a pair of alleles of a character in a hybrid are brought together, and at the time of gamete formation, they segregate or separate. For example, suppose this is the parental characters of first generation. At the time of gamete formation, they separate or segregate. They do not mix. Is it clear? So, according to the law of segregation, when a pair of alleles of a character are brought together, they do not mix with each other. At the time of gamete formation, they separate or segregate. That is called a law of segregation. And these two laws are based on monohybrid cross. And next we can see the third law, that is law of independent assortment. Independent assortment. Before that, I want to tell you about this law of segregation. As I told you, law of segregation is also known as law of purity of gamete, and it is a universally acceptable law. Okay, universally accepted. And here, law of independent assortment is based on dihybrid cross. As I told you, the round green and wrinkled yellow color experiment. And here, he was given capital R for round, then green, small y. Here also small r, then capital Y. Actually, this is round green. I'll write over here, that's better. Round green. Why he was given small letter for green? Because it was recessive character. In the first generation, yellow was dominate. That's why he was given capital Y for the yellow color. Okay. And here this is the wrinkled yellow. So here at the time of gamete formation, this character independent. The segregation of this character is independent of the other. So this is the gamete actually. So here also this is the gamete. So this character is independent of the other pair of character. So we can see what is law of independent assortment. According to the law of independent assortment, when two pairs of a character are brought together in a hybrid, two pairs means round green, wrinkled yellow. When two pairs of characters are brought together, the segregation of one pair of character is independent of the other pair of character. So the separation of one pair of character is independent of the other pair of character. So this is based on dihybrid cross. Okay, is it clear about the laws of inheritance? The three laws by Grigor Johann Mendel. That's all for today.